that. Oh crap, it is time. Well, good morning. This today is going to be the beginning of the innate immunity chapter, and I'm about to post this live stream link to both microbiology sections here momentarily. Micro 1, Micro 2, select right there on the front page module. We'll add an external URL that is this, and give it a title, add item, and then publish. Same thing for section 2. Next semester I'm going to have a single section that includes everyone in the lecture, and the TAs will be able to use a separate section for themselves. So I won't have to do this twice. Here we go. What? I never published. Oh well. No one complained. I didn't publish the lecture last week in lab section 2. Huh. Okay. So those are uploaded, and you see our countdown clock. Please vote if you haven't already done so. All right. You minimize this and that, and close this. And then open slideshow. So here in this chapter, I'm going to introduce the portion of our immune systems that is universal. It's innate. It is non-specific. Okay. So this is our non-specific innate immune response. This does represent our first line of defense and can often prevent infections from gaining a foothold. It is non-specific, so this does not target any specific pathogen. That is the adaptive immune system that is featured in the next chapter. It is innate. These are built-in mechanisms, and this provides an immediate response against potential pathogens. It's not perfect, and this response can and does cause damage to the body at times. So our old nemesis chickenpox, caused by the varicella zoster virus, is highly contagious, and hopefully most of you haven't had to experience that like I did. There was no vaccine available, and well, at least my mother didn't intentionally expose us to it, as was known to happen at the time. Because even before the vaccine, there was a mortality rate of about 3 in 100,000. And those are dice. I mean, why roll them? Particularly now. Sorry, that's kind of loud in there, isn't it? Okay. But the point of this image here is that the inflammation, the rash that is seen, is associated with the body's immune response to the virus. Inflammation is a response mechanism of innate immunity, and usually it helps the body fight off a wide range of infections, but sometimes it does make things worse. Okay, so we have physical, chemical, and cellular defenses. When we talk about physical defenses, we're talking about our physical barriers. Our skin is being primary in that. Mechanical defenses and our microbiome. The microbes that populate us are part of our innate immune defense in that they, even if they don't actively secrete something that fights in potential invaders, the fact that they're already present makes it harder for a potential pathogen to establish itself. Now, for chemical defenses, we have a host of chemicals, some of which are enzymes and our body fluids, antimicrobial peptides that are universal plasma protein mediators, and cytokines, and then mediators that will elicit an inflammation response. The particular immune cells involved here include the granulocytes and agranulocytes, and I'll explain how they were named that way shortly. But first, physical barriers. Now we have multiple types of cell junctions that hold cells together, and primarily these include tight junctions, desmosomes, and gap junctions. Tight junctions are essentially rivets, and desmosomes have intermediate fibers that act like shoelaces, tying two cells together. This allows small materials to pass through the space, and whereas tight junctions prohibit the exchange of materials through that space anyway. And then gap junctions are channels that bridge cells, allowing small materials to pass rapidly between adjoining cells, because gap junctions are channels between two cells to permit communication via signals. So skin. Our skin is a nice thick multicellular layer having three primary layers. We have the epidermis at the outermost, the dermis underneath, and then the hypodermis. So our skin on the outside is dead tissue, and these will continually shed. They make up some proportion of the dust that you have in your home and other spaces. Let's find an actual number for that right quick. Okay, so all right, this is from Live Science. This is a reliable website. I'll put this over here. Said to be comprise dust, but no. Animal dander, sand, insect waste. Insect waste, that's a good thought. Flour, and of course lots of dirt. All right, so it's not. That was an urban legend, and I'm not spreading that. So the majority of your dirt in your house is dirt and insect poop, apparently, and not your own dead skin cells. All right, that was worth looking at. But skin is a fantastic barrier system, because particularly, anything colonized on these dead skin cells also can be sloughed off. Hand washing, helps facilitate that dramatically. Okay, now here we go. Every rose has its thorn. What would a lecture be without a Guns N' Roses reference? So rose gardener's disease can occur, and this is a fungal disease. If it breaches the skin, it's able to infect, and it causes this necrotizing fasciitis type disease here, which again, a while back in lecture, I remember we're talking about gardening. This is one of the consequences of not wearing protective clothing. Potential consequences. I'd have to look up the prevalence of this infection. But the point here is to demonstrate the breach in the skin was required for this infection to happen. You can handle roses all day, and as long as this fungus doesn't get access to a puncture, you're fine. Mucous membranes, part of our physical defense mechanism. This is the mechanical action here, mucous membranes. So here in this SEM, we have the ciliated and non-ciliated epithelial cells from the trachea. We have the mucociliary escalator that pushes mucus away from the lungs, along with all the debris the mucus has trapped. And essentially it moves it all up the trachea to be swallowed. Of course, you voluntarily swallow. You hear the word swallow. Yeah. Here we go. So it is very effective in protecting the lungs, and it is compromised in diseases like cystic fibrosis, and smoking will incapacitate the cilia for a while. And the movement is peristalsis. It moves slowed mucus and other materials. So other endothelia, we have epithelial cells lining the urogenital tract, blood vessels, lymphatic tissue, and others. The blood-brain barrier, which protects the central nervous system, some of the tightest and toughest tissues in the body. Here's a cartoon along with a slide of a goblet cell. These are the cells that will produce the mucus, our mucus. And in this particular type, in the intestinal epithelium. All right, another type of mechanical defense. We have our tears that our eyelid will flush away, the eyelashes to help prevent things from getting into the eye. And urine rinses out our urinary tract, and therefore the urinary tract is typically sterile. Does not make urine magical like some people believe. I don't have a bunch of slides for that in here. I think I've mentioned it before, but there are groups of people that believe in not only saving and then drinking their own urine, but aging it, you know, like 30 days at least first. All they're doing is harming themselves. Like with most woo type beliefs, as long as people don't reject actual medicine, that's fine. If a placebo effect, that does not prevent you from seeking actual treatment or the person, then it's not harmful in that case, except it could just make you a little poorer. But if you shun science-based, evidence-based medicine in favor of these treatments, that's harmful. And the urine people aren't even in that ballpark, because that's flat out harmful. Anyway, okay, the microbiome. So again, this is exclusion by competition. In the vagina, on the skin, in the upper respiratory tract, and the gastrointestinal tract, we have a lot of established normal flora that is able to outcompete any would-be pathogen. And that's not even talking about the normal flora that might actively secrete chemicals that are harmful to would-be pathogens. So anyway, a summary of these cellular barriers. We have our skin, mucous membranes, endothelial cells, which function to deny entry to pathogens. We have mechanical defenses. We shed our skin, the mucociliary sweeping, peristalsis, flushing action of urine and tears, removing pathogens from potential sites of infection, and of course our microbiome, which will compete with pathogens for cellular binding sites and nutrients. Moving on to chemical defenses. So these chemical mediators are endogenous and exogenous. Sebum is an example. It's an oil. It acts as an endogenous mediator by helping seal the hair follicle. However, some genera, Propionibacterium acnes and Malassezia, let's say it that way, are able to degrade sebum as food. Here's a cartoon. Everybody loves cartoons. So the sebaceous glands secrete sebum. It's a chemical mediator that lubricates and protects the skin from invading microbes, though it's also a food source for resident microbes that produce oleic acid, an exogenously produced mediator. So exogenously produced not by us, endogenously produced by us. Okay, the digestive tract. In the oral cavity, we secrete lactoperoxidase enzymes in our saliva that are antimicrobial, along with the amylase and other enzymes. But lactoperoxidases are specifically antimicrobial. In the esophagus, we have mucus. Our mucus contains lysozyme. Stomach, acid. Intestines, we have pancreatic, intestinal enzymes, cryptins, bile, lysozyme, produced by Paneth cells. Urinary tract, our urine is slightly acidic. And in the vagina, lactate, exogenously produced by lactobacilli, which convert secreted glycogen, and this helps to lower pH. Oops, here we go. Eyes. So our tears contain lysozyme and lactoferrin. There was a lab, maybe we should bring the tear lab back. It's a fun demonstration, at least, because you have to make somebody cry, so you can collect some tears and you can put those onto a bacterial lawn and see if there's a clearing. There should be. That's why it's a demonstration. So our ears, our earwax actually has a function that is useful to us. The cerumen contains fatty acids that lowers the pH in our ear canal there to between 3 to 5, which is outside the range that most mesophiles can tolerate. In our respiratory tract, we have mucus. Again, we have that lysozyme, lactoferrin, lactoperoxidases. In our trachea, lung secretions containing surfactant along with lysozymes and lactoferrin plus other chemicals. So an endogenously produced soap of a type. So continuing forward, we have antimicrobial peptides, AMPs. So these are non-specific cell-derived mediators with broad-spectrum antimicrobial properties. So some of these are maintained constantly, or others may be produced in increased amounts in response to a signal of a pathogenic invasion. Now these AMPs can damage membranes, destroy DNA, RNA, interfere with cell walls. And they include defensins, which are produced by epithelial cells throughout the body, as well as by neutrophils and macrophages, secreted or inside host, does damage to plasma membranes. Bacteriocins, exogenously produced by our resident microbiota, often on plasmids that are spread by horizontal gene transfer. So that's the component of our normal flora that's not mechanical but chemical, defensive in nature. Okay, here's this table. As just mentioned, the bacteriocins disrupt membranes. Cathelicidin, licodin, cathelicidin. forgot to pronounce how to pronounce these things sometimes. All right, so epithelial cells. Again, we're disrupting the membranes. We have our defensins, disrupt membranes. Dermacidins from our sweat glands that will inhibit bacteria and fungi, disrupting membranes, membrane integrity and ion channels, and histatins in our salivary glands that will inhibit fungi by disrupting intracellular function. Okay, so another group of chemical defense in our innate immune system are plasma protein mediators. These are acute phase proteins, so these are produced in the liver and secreted into blood in response to inflammatory molecules. And here is a list of some of these. So the C-reactive protein and serum amyloid A, these will coat bacteria, so that process is opsonization, and that will prepare them for ingestion by phagocytes. And we have a couple, ferritin, transferrin, that will bind and sequester iron, thereby inhibiting the growth of pathogens. Sequestering iron in the environment is a very important function for a lot of microbes, particularly in environments that are particularly low in iron, like the open ocean. But when we talk about nitrogen, we talk about carbon sources, we talk about potassium and phosphorus, but iron's pretty high up there in the list of things you need for a living system to function well. So fibrinogen, and that's involved in the formation of blood clots that can function to trap bacterial pathogens. And then mannose-binding lectin, which will activate the complement cascade that will be described. Oh wait, it'll start being described now. Here we go, complement system. So a group of plasma protein mediators that act as both innate, non-specific defense and connect innate and adaptive immunity. So the complement system can be activated by either system. That's why it makes sense we'll talk about it now. So there's more than 30 proteins involved, including the ones designated C1 through C9. They normally circulate as precursor proteins, and they become activated with stimulated or triggered by a variety of factors. And then when activated by alternative pathway, complement system targets pathogens in a non-specific manner. So complement activation. Now when you take cell, you're likely going to have to memorize and describe this in detail. So maybe this will help serve as an introduction to the concept at least. All right, so precursor activation has three mechanisms. We have the classical, alternative, and then the lectin pathway. The classical complement activation pathway is more efficient, depends on antibody production by specific adaptive immune defense. Antibody antigen complex will activate C1. So this is the classical pathway is the interface with the adaptive immune system. So alternative is the spontaneous activation of C3, which involves the hydrolysis of C3 into two subunits A and B. If microbe is present, the B subunit will attach, recruit other complement proteins. Now lectin, similar to the classical, but it's triggered by mannose-binding lectin to carbohydrates on the microbial surface. Lectins, as an acute phase protein, are produced by the liver cells, commonly upregulated in response to inflammatory signals during infection. So here's a little drawing of the pathway. So again, the classical pathway is initiated by an antibody-antigen interaction. There's Antibody is bound to antigen, and that will activate C1, and the lectin pathway interacts as C4, while the alternative pathway refers to C3 being spontaneously activated, and then C3b enters here. And this all results in the formation of membrane attack complexes and various levels of feedback, which is fun. The MAC punches through cell membranes of the invading pathogen, causing them to swell and burst. So the outcomes of complement system activation, we have opsonization. That's where the pathogen gets coated by opsonin, that allowing, it'll help recruit phagocytic cells. And then the membrane attack complex, C6 through C9, allows C9 to polymerize into pores, into gram-negative membranes, the outer membranes, most easily. All right, now cytokines. So cytokines are molecular messengers that have effects on every, type of cell signaling mechanism here. So the same cell can be activated by the release of cytokines, and that's autoinduction, and so it's autocrine. Paracrine is where the secreting cell is communicating with a nearby cell that has a receptor. And then endocrine, of course, is when the cytokine travels the immune system or whatever signaling molecule or hormone. Well, those are hormones. So the cytokines are able to activate distant cells that are able to respond. Cytokines are soluble proteins that act as intracellular signals. Okay, yeah, that's kind of clear. All right, there are three important classes. We have our interleukins, our chemokines, and interferons. The interleukins are not exclusive to leukocytes. They are produced by and stimulated by a variety of cells unrelated to immune defense. Chemokines, chemotactic factors that recruit leukocytes to the sites of infection, initiated by tissue damage and involved in inflammation, specific to the types of cells recruited. Interferons, immune signaling molecules important in viral defense. A couple types of interferons I'm going to describe now. So we have type 1, and that's alpha and beta included in that group. These are produced and released by cells infected with virus that stimulate nearby cells to stop producing messenger RNA, destroy RNA, and reduce protein synthesis. The objective being to inhibit viral replication and slowing viral spread. And these types of interferons will also stimulate various immune cells to more aggressively attack virus-infected cells. Type 2 interferon gamma is an important activator of immune cells, alert neighboring immune cells to attack. Here's an interferon cartoon. So here they're being generated by an infected cell, and they can signal a neighboring cell to undergo apoptosis if they're infected, or to destroy RNA and reduce protein synthesis if they're uninfected, and also function to activate immune cells. Okay, right. I guess that's important to point out. So the interferons don't save the cell, the host cell. The host cell that is secreting the interferon is going to die. I mean, if nothing else, through an autocrine induction, right? Induce that apoptosis. All right. Next, we have our inflammation eliciting mediators. Histamine, we've all probably heard of. This is a pro-inflammatory compound that are released by mast cells and basophils. Histamine receptors are found on a variety of cell types, and they of cells that mediate pro-inflammatory events such as bronchoconstriction and smooth muscle contraction. We have the leukotrienes. These are produced by mast cells. They are lipid-based pro-inflammatory mediators produced from the metabolism of arachidonic acid in cell membranes of leukocytes and tissue cells. More potent, longer lasting than histamine, can induce coughing, vomiting, and diarrhea, which could be important things to induce, right? And then we have bradykinin. This mediator contributes to swelling, to edema, which again has a purpose. All right, here's a summary of chemical defenses. And so our sebum, oleic acid, lysozyme, acids, well, stomach, urine, vagina, ear, digestive enzymes, bile, lactoferrin, transferrin, sequester that iron. That's important. Surfactant, defensins, bactericidins, dermacidins, cathepsidins, histatins, antimicrobial amps, antimicrobial peptides. Then we have our plasma protein mediators, including our complement, our acute phase proteins, and then 3 types of cytokines, and then our histamine, leukotrienes, prostaglandins, which promote inflammation and fever. I did not mention them on the other page, and bradykinin. Okay, so that leaves us third category of cellular defense. So 3 types of what was originally called formed elements. They're all derived from pluripotent hematopoietic stem cells, HSCs. Hematopoiesis, the differentiation process of blood cells. Vast majority become erythrocytes, red blood cells. The ones that become leukocytes, there's 2 classes, granulocytes and agranulocytes. Here's the cartoon for this. Hopefully that looks, it probably doesn't look good at all in this video. Okay. Okay, so some stem cells remain. That's important to point out. We can make lymphoid stem cells, myeloid stem cells. The myeloid stem cells can differentiate into the megakaryocyte, erythrocyte, the red blood cell, mast cell, those are an important immune component, myeloblasts, which can differentiate into basophils, neutrophils, eosinophils. Yeah, and then we have macrophages and dendritic cells coming from monocytes. Meanwhile, the lymphoid stem cell can differentiate into natural killer cells, the NK cells, and the small lymphocytes, so the T lymphocytes, B lymphocytes, and then plasma cell. These are important components of the adaptive immune system as described in the next chapter. Okay. So, the granulocytes versus the agranulocytes. Those were named as such based on the number of granules and the size of the granules that could be seen inside the stained cell initially. So red blood cells, this is the vast majority of our blood. Fortunate for us, our blood has a lifespan of about 120 days. As you know, the red blood cell ejects its nucleus to carry more hemoglobin. So it's oxygen, carbon dioxide, right? So leukocytes in general, these have an obvious dark staining nucleus. So if you're looking at a blood smear, that's an easy way to tell the difference. They're a lot fewer in number, and they all function in body defense. They're able to exit capillaries and move into tissues. Lifespan of usually a few hours or days. Of the granulocytes, the majority are neutrophils. The nucleus, lobes, the number of lobes increase with age. They have pale lilac granules. They are phagocytic, particularly effective against bacteria. They release cytotoxic chemicals from the granules. Most common leukocyte. Oops. Wait, am I going to go into... Oh, whoops, I forgot I did that. Wait, let's scan ahead a little bit. Oh, we're going to go through a description of all of them anyway. Maybe I'll save that. 33 minutes, a little bit short, but I think I'll wait here and go into this on Wednesday in person. And that should, yeah, that'll work. Anyway, have a good day, and that's it. Remember to vote. Hopefully you already have if you live away from home. All right, if you can vote and all that. Oops, broadcast.